Okay, let's start. So we have already completed the structure, mainly the anatomical structure of the lens. Now I'm going to talk about the physiology of the lens. So in physiology of the lens, the biochemical composition of the lens, it contains around 65% water, 34% protein, and rest 1% lipid, carbohydrate, ascorbic acid, glutathione, amino acid, and inorganic ions. Water content of the lens. Lens is relatively dehydrated organ. Dehydration is maintained by the active sodium pump mechanism within the cell membrane of the epithelium and each lens pipe. 80% water is free and rest 20% is bound water. In normal lens, there is no significant alteration in hydration with the age. So what they're telling over here? First, they say that the composition, biochemical composition basically of the lens. So what are the things are there? Mainly water, the maximum portion is water. Then we have protein. Then have some lipid, carbohydrate, ascorbic acid, glutathione, amino acid, and inorganic ions. That's a very, very limited one. That is 1%. So the main role is to maintain hydration of the lens, which is related to our transparency of the lens actually. So lens is basically dehydrated state. And it's maintained by what? Maintained by the sodium pump mechanism in the epithelium and also the each lens pipe. So that you have to remember that the hydration is maintained by the sodium pump mechanism. And in normal lens, there is no significant alteration in hydration. If it is normal state, the lens is in normal state, so without any abnormality, so there is no question of any changes in the water content on the lens with the age. Protein content higher than the any organ of the body. So they say that the content of protein is higher than any organ in the body. They have basically two types of the protein present in the crystalline lens. One is a soluble fraction or soluble protein, you can say, called as crystalline. That's why the term came over there, crystalline lens. We usually say the crystalline. Instead of lens, we say crystalline lens because of the soluble proteins. And we have again another one is the insoluble proteins. They're called as albuminide. Young lens fiber contains more soluble protein or fractions we call than the older fibers. So remember for short question this one. So young fiber contains more soluble fraction compared to older fibers. Soluble fraction or soluble protein, that is called crystalline protein, are three types. Alpha crystalline around 31.7%, beta crystalline 53.4%, and again gamma crystalline, that's 1.5%. Insoluble fractions or albuminide having around 12.5%. And other protein, we have mucoprotein and nucleoprotein. We have respectively 0.8% and 0.07%. Very less number. Coming to the soluble protein. Structural protein, C-max, build the refractive fibers. So this is another important aspect of the lens fiber because all the light passes through the mass structure of the lens is definitely the lens fiber. So the light passes through that. So definitely this is the major important, one of the important parameters. Synthesis takes place in the equatorial part of the lens and other surface of the lens. Alpha crystalline having highest molecular weight. Alpha crystalline is a polymer made by 50 monomers. Beta crystalline have the high thiol content and disulfide linkage. Gamma crystalline composed of monomers only, and gamma crystalline level is high in nucleus than the cortex. So another important aspect over here. So the short question wise, the gamma crystalline level is higher in the nucleus compared to cortex region. Remember that. Coming to insoluble protein, that is called albuminide. Amino acid composition is similar to the alpha crystalline. Most albinoid is urea soluble and appears to be derived from the alpha crystalline. So what you can see over here, that albinoid is basically urea soluble and appears to be derived from the alpha crystalline protein. Other protein like glycoprotein we have, the protein bound with sugar with covalent bond. That's called glycoprotein. Nucleoprotein, phosphoprotein, lipoprotein, etc. present in the lens. The lens protein are organ specific. An individual can become sensitized to his own lens protein. This is another factor. Okay? Because we can see so many proteins are present in the lens. That's why they say that is the highest number of protein present in the lens compared to other organ in the body, whole body. So sometimes it can harm the scientific structure of the lens. Sometimes this one of the protein denatured due to some malfunctions or disease or some external effects. So that protein gets denatured and cause problem on that. So, but it's definitely a person to person, it varies. Amino acid. Lens contain all amino acids except tryptophan, cysteine, and hydroxyl propylene. Amino acid actively transported from the aqueous to the lens. So the source of amino acid is from the aqueous. Amino acid concentration of the lens is not affected by the age. Another important thing they say that the amino acid content not affected by the age. Carbohydrates, basically glucose and fructose. Glucose level of the glucose in the lens is 1/10 of aqueous humor, where the glucose concentration has been found to be 100 mg. Fructose produced from the glucose. Lipid, total lipid of the human lens amount about to 2.5% of the weight. The main substance of the cholesterol and various phospholipids. So the lipid basically we have cholesterol and various phospholipids. Around 65% of the lenticular lipid are bound to the protein. Feldman have demonstrated that in cataract, the concentration of free lipid increases and lipoprotein decreases. So another theory actually, Feldman has given that in cataract when the opacification of our crystalline lens is due to the age or various factors, so that called as cataract. So there they have observed that the concentration of free lipids increases and the lipoprotein decreases. Glutathione present in the lens varies from 3.5 to 5.5 mg weight. Its amount altered with the age. It is a tripeptide and consists of 3 amino acids, that is glycine, cysteine, and glutamic acids. So the 3 types of amino acids is present over there. That's why it's called tripeptide. And what are the names of glycines, cysteine, and glutamic acid? Glutathione contributes the redox system of the lens microenvironmental factor. More than 95% glutathione is reduced on the present. Ascorbic acid. The mean value of the ascorbic acid in human lens is around 30 mg of the weight of the lens. It is neither synthesized or not actively transported in the lens. The precise role of the ascorbic acid in lens metabolism is not well established. So we don't know what are the exact functions of the ascorbic acid in lens metabolism. So coming to the metabolic activity of the lens, this one is very important actually. You have to remember this one. So glucose metabolism. Lens require energy in the form of ATP. We know that one already read in during the cornea. So in these various metabolic activities, definitely we have so many 4 or 5 types of metabolic activity. So this energy is achieved by the glucose metabolism, and 10 to 20% ATP used in the protein synthesis. Rest ATP used in the transport ions, amino acid, maintenance of the lens hydration and transparency. Most of the ATP used in the epithelial level and 3 to 4 milligram of glucose per day is utilized for the lens. So we get an idea about that glucose metabolism. Really. So two most important things over here. You just have to remember that. First is that the energy is definitely by they're getting by the glucose metabolism. Second, the 10 to 20% only used for the protein synthesis. Rest of the thing only for the to maintain the dehydration and transparency of the lens. Rest almost 80%. Glucose deprivation in the lens can cause utilization of other sources. The ATP orbital fructose become hydrate and thus the loss of transparency. So if the any by any conditions, if there is a deprivation in the lens, glucose deprivation, because as I said, you need a 3 to 4 milligram glucose per day to maintain the regular or normal metabolic activity. So if it's not there, then definitely there is other sources, but that won't be sufficient by the other sources like sorbitol, fructose, and lens become hydrated also, because we know the lens is dehydrated state. So if it's hydrated means the definitely accumulation of water only, and when it causes due to the malfunction of the metabolic activity, definitely. So that's why they lost the transparency. If the accumulation of water is there, any transparent organ, so definitely they lost their transparency. So there are 4 pathways there which helps to metabolize the glucose: anaerobic glycolysis, Krebs cycle, HMP shunt, and sorbitol pathway. Same like our corneal metabolism pathway. Here the diagram shows that various what I mentioned over there, that 4 pathways are there. So we can see that what is happening over there. In a very nicely they have put in a diagram chart. So glucose with the enzyme called hexokinase, they form the glucose 6-phosphate. From there, glucose 6-phosphate dehydrogenase, the enzyme 6-phosphate glucose gluconate they form. From that, NADP to form the NADPH. And from there, the hexose monophosphate shunt take place, the metabolism pathway, and they produce the pentose phosphate. Then from there, they produce the fructose 6-phosphate, and fructokinase enzyme converted fructose 6-phosphate to the fructose. In the other, if we see glycolysis pathway, so from the glucose 6-phosphate, the glycolysis pathway, it directly to fructose 6-phosphate. Then from the phosphofructokinase, it's become fructose 1,6-diphosphate, and from there it converted to the pyruvic acid, and that is again from the Krebs cycle. So lactate dehydrogenase enzyme, they call pyruvate. This confirm that in the Krebs cycle. So this is the, again another one is the direct pathway is the aldoreductase, that enzyme help to NADPase to NADP in the orbital pathway, that is, and from there they produce the fructose. Polyol dehydrogenase enzyme works over there, and then they put the fructose over there. So this is the whole pathway actually. This is the whole pathway where the metabolism take place over there, and what is the end product? There's the lactate over here. The end product, basically the lactate. So as you see that 4 pathways, all are here. They have nicely described that. And what are the enzymes also actively participating, also mentioned over there. So from glucose to glucose 6-phosphate, hexokinase enzyme is helping. From the glucose 6-phosphate to 6-phosphate gluconate, who is helping? That is a glucose 6-phosphate dehydrogenase enzyme. And where the NADP is a formation of NADPH over there. And for glycolysis, from the glucose 6-phosphate, this become the fructose 6-phosphate. And with the help of the fructokinase enzyme, it becomes fructose. So there's that. And again, the phosphofructokinase enzyme from there to fructose 6,1,6 diphosphate, and then from that is pyruvate. Pyruvate again, it's a lactate dehydrogenase enzyme converted to lactate. This comes under the Krebs cycle. So you see that the whole chapter is basically related to the metabolism of the glucose. Coming to the multimillerage, the synthesis from the free amino acid, which are actively transported into the lens from the aqueous. Peptides form from amino acids with the requisite ATP and RNA template. Rate of the protein synthesis is slow in nucleus than the other parts of the lens. Permeability and transport mechanism. There's 2 types of transport mechanism on the present in the lens. One is the active transport mechanism and the passive transport mechanism. Active one is they use the ATPs, and 90% transport takes place in active transport. The transport amino acids, potassium, taurine, inositol, extrusions of sodium ion. In passive transport, what are the things that occurs across the lens capsule for water, ions, waste product of metabolism, like lactic acid, carbon dioxide, etc. So passive transport product, basically lactic acid, but the waste product of the metabolic activity that transported through the passive transport. This consists in permeable to low molecular weight compound and restrict the large colloids. What are an electrolyte transport? The cation form. The functioning at level, the anterior lens epithelium, with the help of the ATP and sodium, is the actively extruded and uptake of the potassium takes place. This process is active transport stimulate the passive diffusion called as pump and leak theory. So what is happening over there? Cation pump? Cation pump basically it takes place in the lens epithelium. So we have only one epithelium, the anterior surface. So there, it's already said that outer and electrolyte transport. So definitely, with the help of ATP has to help because energy. So actively, that sodium and potassium exchange has to take. This process, active transport, stimulates the passive diffusion called as pump and leak theory. So this takes place because of the this goes out and comes in. Well, definitely the passive diffusion technique. Lens as osmometer. The lens considered as a single giant cell which swells up in the hypertonic medium. Increase in sodium and potassium increase osmolarity and cause the lens swell and loss to transparency. Here you can see that one, that the 3 types of active transport, that the pump mechanism, that is diffusion or the leak. That's why the theory came up with that. And the S is the stands for the synthesis. So red color one, we can see the active transport for the sodium and potassium. We can see the transportation, and the blue color one, the diffusion. So diffusion means definitely from the higher concentration to the lower concentrations. So here you can see the chlorine, glucose 6. Here is 1. So definitely is goes in. Lactic is also for the same from the 14 to 17. So, and synthesis takes place for the proteins. Lens transparency, it depends on the few factor. Definitely avascularity of the lens. Yeah, lens is avascular. There's no blood vessels on the lens. We know that one. Single layer of epithelial cells, the front surface, the anterior surface, that is the very important one. Semi-permeable nature of the lens capsule. Yes, they won't allow everything to enter to the lens. Tracity of the highly packed lens cell. Yeah, we have seen that tightly packed the lens fibers already. Characteristics arrangement of the lens protein. Yes, definitely that's important. Form mechanism of the lens fiber. Auto-oxidation, reduce the glutathione, keep the lens protein reduced state, and provide the integrated cell memory.